Hola, buen día. My name is Talia, and I am a student at Universidad Juárez Autónoma de Tabasco. In the present study, we identified the taurine biosynthesis pathways and the taurine transporter present in early ontogeny and in different organs of tropical gut. Taurine is a natural sulfur compound found in most tissues and organs of vertebrates. It is known that taurine participates in a wide variety of functions such as skeletal muscle development and the formation of bile, retina, and brain. This taurine can be acquired through food absorption in the intestine or by the novo synthesis from his methionine cysteine precursors. In fishes, 3 different pathways and participating genes coding for proteins have been described. In pathway 1, the cysteine sulfinato-dependent pathway, we have the gene cysteine deoxygenase type 1 and cysteine sulfinic acid descarboxylase genes. In pathway 2, the cysteine acid pathway, we have genes cysteine deoxygenase type 1 and glutamic acid descarboxylase gene. And in pathway 3, the cysteamine pathway, we have the 2-aminoethanethiol deoxygenase gene. It is also known that taurine transporter is the responsible for the produced taurine transportation and that the predominant pathway will depend on the species, type of tissue, stage of development, eating habits, and the environment in which they live. However, we found there is no information regarding the different pathways or genes involving taurine biosynthesis in the lepidosteiformes order. For the present study aims to determine the expression of genes involving taurine biosynthesis and its transport during early life cycle stages and identify the pathways present in different organs of tropical gut for a better understanding of its mechanism. So, samples for the present study were obtained from an induced breeding spawning at the Laboratory of Physiology in Aquatic Resources, where the care of the recently hatched organisms lasts 31 days as we can observe in the timeline. At the end of the 31 days, we dissect organisms to obtain individual organs. Genes involved in taurine biosynthesis, its transport, and reference genes were retrieved by bioinformatics research in the supplemental material of tropical gut transcriptor to design the primers used in the qPCR. As we observed in the workflow, that was what we did to obtain the expression. So, as results, we found that the relative expression of all genes was detected at hatch and was broadly expressed in all genes. Therefore, results indicate that taurine can be synthesized endogenously before first feeding in tropical gut, inferring that taurine is maternally derived. This can be corroborated by the relative expression of hatch of taurine transporter. As we can observe in the graph, it was detected at hatch and broadly expressed in all ages. We also found that for organs, the highest expression level of taurine transporter were found in eye and was barely detected in brain and stomach. So, to determine the predominant taurine pathway occurring during early ontogeny and type of organ in tropical gut, we construct heat maps using relative expression values obtained in qPCR. We observed that at hatch, contrary to what has been found in other species, where the cysteamine sulfinato-dependent pathway is the main pathway in tropical gut is the cysteamine way. We observe cysteine sulfinato-dependent pathway mainly occurs at 5 days after hatching, while cysteine acid pathway is predominant at 3 and 17 days after hatching. However, the main pathway for taurine biosynthesis at the different days after hatching is the cysteamine pathway. Moreover, in figure 5b, we can visualize that the principal pathway is organ-dependent. We observe cysteine sulfinato-dependent pathway majorly occurs in the liver, followed by skin, muscle, and intestine, where cysteine acid pathway only occurs in the eye and brain, which is very consistent with other studies in mammals such as cats, bovine, and rats. However, cysteamine pathway occurs in most organs, mainly in the eye, liver, and skin. This result suggests embryos, larvae, and juveniles can synthesize taurine through the 3 pathways, and that cysteamine pathway is the main taurine pathway at different early ontogenetic stages. We also found that taurine biosynthesis is organ type dependent, indicating that the predominant is cysteine sulfinato-dependent pathway in the liver and only cysteine acid pathway occurs in the brain and eye, and that the cysteamine pathway mainly occurs in the eye. However, further research is needed to determine the effect of taurine at different physiological and nutritional levels. Thank you.